Hey, ladies and gentlemen, today we are going to work on a lab. However, I do not actually own the materials in order to do this lab, so we are going to be doing this in a virtual setting. Problem is that the technology used, called flash animation, has gone obsolete. So, while I still have access to it, I'm going to record myself going through it, and we're going to use that until I can come up with a suitable substitute for this program. All right, so let's jump into this. The first thing you should do is jump back into Google Classroom and find a Google Doc that looks like this. Now, this is not the Google Doc at the time of recording. I did not make that Google Doc. This is an older Microsoft Word file, but it should look similar. I do not know if I'm going to include all of this stuff in. All of this was to make sure you were following the directions by filling in the blanks as you go. This time around, I'm going through the directions. So we will see if I decide to put this into your Google Doc or not. So here we have your virtual lab. This is going to be a point and click thing that you're going to follow along with me with. You are holding a small plastic tube with clear liquid in it. You've been told that the liquid contains DNA strands of several different lengths. Your job is to figure out what those lengths are. How will you do it? There's your DNA. If the DNA strands were as big as your shoelaces, you could sort them by hand in the groups and measure them. But DNA strands are molecules so tiny that you can't even see them under most microscopes. Is there a way to sort and measure DNA strands in your tube even though you can't see or touch them? There is. It's called Gel Electrophoresis, pronounced Electrophoresis. All right, guys, I'm going to back up for just a second. All right, your DNA actually does naturally chop itself up into pieces, 46 pieces called chromosomes. All right, that's what we're going to do. We are going to chemically chop our DNA into these pieces and then separate them out. Your pieces will always separate into the same lengths, and the DNA that you got from your dad will be the same length as yours. The DNA you got from your mom will be the same length as yours. So your DNA will look like half your mom, half your dad. So let's see how this is done. Scientists use gel electrophoresis whenever they need to sort DNA strands according to length. This technique is also useful for separating other types of molecules like proteins. How does this work? The gel is a filter that sorts the DNA strands. It's like a sponge made of jello with many small holes in it. We place the DNA samples into holes at the end of the gel. Right? So this is where all the DNA is right now. Electrophoresis is how we push the DNA strands through the gel filter. By adding an electric current, we can make the DNA move. Short strands move through the holes in the gel more quickly than the long strands. Over time, the shorter strands in the sample will move further away from the starting point than the longer strands. So you see the short strands far away here, and then the longer strands are closer to the beginning. DNA strands at the same length will move at the same speed and end up grouped together. In this way, the DNA strands in the sample sort themselves. Staining the sorted groups of DNA make them visible to the naked eye. Normally we can, I'm not reading here. Normally we can't see the DNA, so we see the stain or the dye that we add to it. Although we can't see a single DNA strand, we can see large groups of stained DNA strands. These groups show up as bands in the gel. Now it's your turn to run a gel. That's scientist slang for gel electrophoresis. Follow along with the steps shown above. 1. Make the gel. 2. Set up the gel apparatus. 3. Load the DNA sample into the gel. 4. Hook up the electrical current and run the gel. And 5. Stain the gel and analyze the results. So let's go with step 1, make the gel. And by the way, guys, this is how we would actually do it in a real lab setting. I just don't have the materials. That's why we're doing it this way. Here's what you will need to make the gel. Powdered agarose, a buffer, a flask, a microwave, the gel mold, and the gel comb. Put a small amount of the agarose into the flask. Agarose is a dried powder similar to gelatin, but made from seaweed. Add some liquid buffer to the flask. The buffer is a saltwater solution that will let electric charges flow through the gel. That's important to remember, guys. Electricity runs better through salt water. We've loosely placed plastic wrap over the top of the flask to prevent the liquid from boiling over. When the microwave, open the microwave by pressing the open button. Place the flask containing the buffer and the agarose mixture into the microwave. Heat the mixture until the agarose melts into the buffer. We've removed the plastic wrap from the top of the flask. Pour the melted agarose mixture into the mold. Notice that the mold has tape on each end to hold the melted agarose in. Place the comb into the gel on one end. The notches in the gel mold hold it in place. Let the gel cool and solidify. This usually takes about half an hour, but we'll speed up the clock for you. As the gel cools, tiny holes will form in it. Carefully remove the comb, leaving empty wells for the DNA samples. Now it's time to set up the electrophoresis box. You will need the gel you just made, an electrophoresis box, and another bottle of buffer. Pour the buffer into the electrophoresis box. Remember, salt water helps electricity go. Place the gel, still in the mold, into the electrophoresis box. Sorry for that pronunciation. We have removed the tape from the ends of the gel mold. The gel should be just barely submerged in the buffer. You're ready to load the DNA sample into the gel. Here's what you need. Loading buffer, right here. The tube of DNA, here's your DNA samples, and the DNA size standard, right here. This is a sized out DNA. The scientists already know how big the DNA is here. They're going to use it as a ruler to measure this DNA. A micro pipetter. The electrophoresis box contained the buffer and the gel, and some pipette tips. Load the DNA sample into the gel. With a clean pipette tip, use the micropipetter to suck up some of the loading buffer and then add it to the DNA sample. DNA samples are prepared in a clear liquid solution that would be hard to see if you tried to load directly into the well. The loading buffer contains a dye that makes the sample easy to see. It is also slightly goopy. This makes the DNA sample thicker so that it will drop into the well instead of floating away. The DNA size standard already contains the loading buffer. Next, you will use the micropipetter to transfer the DNA sample from the tube into the well of the gel. First, suck up some of the DNA sample into the pipette tip. Next, you will eject the DNA sample from the pipette into the first well of the gel. Nice job! In real life, loading the sample into the wells takes some practice, so don't be disappointed if you miss your target the first few times. Using a clean pipette tip, use the micropipetter to suck up some DNA size standard. Transfer the DNA size standard into the next empty well. The DNA size standard contains DNA strands of known lengths. Running it on the gel will give you a reference by which to estimate the lengths of DNA strands in your sample. Like I said, we already know how big this DNA is. We're going to use it as a ruler to measure this DNA. It's time to turn on the electricity and run your gel. When you turn on the power, the black end will generate a negative charge. The red end will generate a positive charge. Together, they will pass the current through the gel. DNA has a negative charge. To move the DNA through the gel, you must put the black cord, the negative charge, closest to the wells. By the way, electricity moves from the negative side to the positive side. So if the negative charged DNA is over here by the negative charged wire, that electricity will move this way, dragging the DNA with it. Plug the black cord, if it'll let me. Plug the black cord from the electrophoresis box into the matching outlet of the power supply. If it will let me. Oh. That's right. Now click the location on the power supply where we want to plug the red cord. And turn on the power supply. Your gel is off and running. Let's look into the gel box and see what's happening. Check for tiny air bubbles coming out of the electrodes at both ends of the electrophoresis box. These bubbles are your proof that the current is running. So here we got the negative charge close to the wells, and the electricity is going from the negative charge across over here to the positive charge. And that's going to drag the DNA with it across the gel. Repelled by the negative charge, the DNA moves through the gel towards the positive charge at the other end. Short DNA strands move through the holes in the gel more quickly than the long strands. Over time, the shorter DNA strands will migrate farther from the starting point than the longer strands. We can't actually see the migrating DNA bands, gray, but we can see the blue dye from the loading buffer as it migrates. You finished running your gel. We've taken the gel mold with the gel in it out of the electrophoresis box. The time has come to analyze your samples and estimate the length of the DNA strands. First, you need to stain the DNA in your gel using DNA staining solution. The stain is a chemical called ethidium bromide, which binds the DNA and shows up under a fluorescent light. Though we can't see the single DNA strands, we can see large groups of stained DNA strands. These groups will show up as bands in the gel. Drag the gel out of the mold and put it into the DNA staining solution. It takes about half an hour to stain the DNA in the gel. We sped up the clock for you. Remove the gel from the staining solution and place it on the UV ultraviolet light box. Turn on the UV box. Ultraviolet UV light from the box can damage your eyes. If you do this in real life, be sure to wear protective goggles. Now you can determine the approximate lengths of the DNA strands in your sample. Compare the bands of the DNA sample with the bands of known lengths from the DNA size standard. Enter the estimated length in base pairs, BP, for each band in your DNA sample. Electrophoresis cannot tell us the exact DNA lengths of DNA strands, but it's a good estimate. So give it your best guess based on the DNA size standards. Press forward when you are finished and check your answers. I am not going to put these numbers in for you. You are going to put them in here yourself. All right. So here's your size standard that we use as a ruler. This is 6,000 base pairs, 5,000 base pairs, 4,000, 3,000, 2,000, 1,000 base pairs. Our first DNA sample is the same level as this one at 6,000. The second one is in between 3,000 and 4,000. And the third one is in between 1,000 and 2,000. So it's up to you to come up with these numbers and put them here on your paper. There are 8 questions. So when you're done, I will be able to see them on Google Classroom, and I will see you next time in class. If you have any questions, email me or write me on Google Classroom. Have a nice day.